Assalamu alaikum. This is a description of the gastroesophageal junction together with the pyloroduodenal junction and the duodenum. This is the gastroesophageal junction. How can I know it is a junction? Because there is a change in the appearance of the epithelial lining from one side to the other. Also, you can see that there is a change in the thickness of the muscle from one side to the other. Starting with the epithelial lining in this area, I can see that it is a thick epithelium and it is not showing openings, numerous openings like this side. In this side, I have a thin lining because this is the one layer of nuclei, and I have numerous openings, white openings in this epithelium. So this is the gastroesophageal junction because this is the side of the non-keratinized stratified squamous epithelium, and this is the side of the simple columnar and showing the gastric pits. Here we have the lamina propria showing some ducts of the esophageal glands, which pierce the muscularis mucosa, so it is not so evident here but very evident there. Here we have the esophageal glands in the submucosa, which are mucous compound tubuloalveolar glands, and their ducts pass through the muscularis mucosa to open in the epithelium and to open and to pour their secretion in the lumen. Here we have the muscle thick relatively in the stomach or in the fundus compared to that of the esophagus, because we have an additional inner oblique which will appear oblique in the longitudinal section or transverse section. Of course, the junction is a longitudinal section, never a transverse. To take the changes from one area to the other, you should take a longitudinal section and not a transverse. Transverse will pass in only one layer or one level. Here we have the appearance of the circular. It will appear circular, and the longitudinal will appear in the form of a spindle. So here the appearance is the reverse to that in the transverse section. And then finally we have the serosa. Here we have also a junction. We have a change in the epithelial lining, non-keratinized stratified squamous epithelium, thick, solid, not interrupted by the pits, and here we have thin epithelium and it is interrupted by the gastric pits. Here we have the lamina propria, interrupted muscularis mucosa, esophageal glands, mucous compound tubuloalveolar, muscularis mucosa, connective tissue devoid of glands here, but here the glands in the submucosa, as we have said, inner oblique, middle circular, outer middle circular and outer longitudinal, هنا inner circular and outer longitudinal. Here we have the non-keratinized stratified squamous epithelium, as you see, very thick epithelium at the esophageal side, and here we have a simple epithelium formed of one layer of cells with basal oval nuclei, and the top of the cell is very vacuolated and they line also the pits. And then we have the gland. The glands are very crowded, occupying the lamina propria, making the connective tissue very limited in this area but evident in the other side of the esophagus. Now we will move to the quiz. Here we have this junction. The epithelium changes from non-keratinized stratified squamous, pointed to by the red arrow, to simple columnar epithelium, pointed to by the black arrow, with the appearance of the fundic or the gastric pits, pointed to by the blue arrow, and the gastric fundic glands occupying the lamina propria, pointed to by the green arrow. It's a junction between the esophagus and the stomach, and this junction is known as the gastroesophageal junction. Second slide showing also the gastroesophageal junction, gastroesophageal. This type is called of junction is called gastroesophageal junction. The side A is the side of the esophagus. Why? Because of the presence of the non-keratinized stratified squamous epithelium. Side B is the side of the fundus of the stomach. Why? Because it is lined by simple columnar epithelium and it shows the gastric pits. Red arrow denotes the gastric pit. The star denotes the lamina propria of the mucosa layer occupied by the gastric fundic glands. Here we have the gastroesophageal junction. Also, the glands outlined by the blue circle here are the esophageal glands. They lie in the submucosa layer. Their type is mucus compound tubuloalveolar. The glands outlined by the red circle are called the gastric fundic glands. They lie in the mucosa layer. Here, this is the picture for the section in the small intestine. Here we have this fold, which is a circular fold. Remember that the stomach has longitudinal folds called rugae. Here we have in the small intestine circular folds, which are called plica circularis. What is the function of this circular fold? Is to increase the absorption of digested material 3 times. Here we have in the mucosa finger-like projections, which are called villi. And we have simple tubular glands, which are the intestinal crypts of Leberkin. The villi increase the surface area 10 times. So the plica circularis 3 times, villi 10 times. Also, the villi are lined with cells or covered with cells. These cells have microvilli or brush border that increases the absorption 20 times. So remember 3, 10, and 20. Here we have the muscularis mucosa at the end of this mucosa, and then we have the submucosa connective tissue and the musculosa formed of inner circular and outer longitudinal. Here we have the intestinal villus. It is a finger-like projection containing core of connective tissue from the lamina propria, containing blood vessels, nerves, and a central lacteal or a lymph vessel. Here we can see 2 evident types. We have the columnar absorptive cells or enterocytes having this as eosinophilic brush border, which increases the rate of absorption 20 times. The villus itself increases the rate of absorption 10 times. Here we have this cell, it's columnar with basal oval nucleus, and it is provided with eosinophilic brush border, which is nothing but microvilli. And here we can also see clearly the goblet cells, which is having the shape of flask اللي هي which having the shape of case يعني. Here we have the upper part which is expanded, full of the mucus which dissolves during preparation of the section, and the lower cylindrical part containing the triangular nucleus. And they are present here in the villus as well as in the crypt, together with the enterocytes or the columnar absorptive cells. Here we have the brush border of the columnar absorptive cells, and this is the lamina propria. Also, we have the villus finger-like projection containing core of connective tissue that contains blood vessels, nerves, and the lacteal. Here we have the goblet cells, and here we have the enterocytes or simple columnar absorbing cells with characteristic brush border which is acidophilic to increase the absorption 20 times. And the goblet cell, of course, secrete the mucus for protection and lubrication. Also, we have here the picture for the villus, which is projecting on the surface, and the crypt, which is an invagination here in the lamina propria. It is a simple tubular gland, like a test tube. Here, this crypt secretes the intestinal juice. Here we have the characteristic Pannis cells at the base of the crypt. The Pannis cell here is pyramidal with a basal rounded nucleus, and the apical part of the cytoplasm is full of acidophilic granules. These cells are very characteristic for the crypt of the small intestine. It is not present in the crypt of the large intestine, and it is responsible for the secretion of the intestinal enzymes as well as the lysozyme, which is an antibacterial material to protect you against the bacteria which is present in huge amount in the lumen of the intestine. So this downward invagination is the crypt, and these are the Pannis cells. Here it is the crypt. Here we have the villus. This is the crypt. So these are three crypts, and these are the Pannis cells. This is the blood vessel, of course, in the lamina propria, and this is the muscularis mucosa. Here we have the core of lamina propria inside the villus. In the other picture, you can see it is a simple tubular gland having Pannis cells at the base of the gland, and the cells are characterized by the apical acidophilic granules. Don't forget it is having the same shape of the peptic cell of the stomach. It is pyramidal with basal rounded nucleus, but here we have evident acidophilic granules or eosinophilic granules here in the apex of the cell. Also, don't forget that we have here goblet cells also in the crypt, together with the columnar absorptive cells. The columnar absorptive cells, of course, have two important functions. They are responsible for the absorption in addition to complete the digestion of the carbohydrates and the proteins. Here we have the first part of the small intestine, which is the duodenum. This is a small intestine. Why? Because I have a villus and a crypt. We have projection above the surface and downward invagination, which is the crypt. This is the projection and this is the invagination. So we have villi and crypts. Villus and crypt. Villus and crypt. Here we have the muscularis mucosa ending the mucosa, and in the submucosa I have a lot of mucus glands, which are mucus compound tubuloalveolar glands, and they secrete this mucus for protection and lubrication, in addition to the urogastrone, which inhibits the secretion of the HCl by the stomach. Here we have, after that, the musculosa formed of inner circular and outer longitudinal. The presence of the mucus glands is only in the duodenum, and they are called Brunner's glands. So the presence of these glands denotes that this is the duodenum, not ileum, not jejunum. Here again, it is a section in small intestine because I have projection on the surface and downward invagination. Here a projection, and this is the invagination. This is the villus and this is the crypt, and we have the connective tissue of the lamina propria. Here the muscularis mucosa, and we have here the Brunner's glands whose ducts open in the base of the crypts of the mucosa, and as we have said, it has two functions: the production of the mucus and also the inhibition of the gastric HCl secretion. Here we have the musculosa, inner circular and outer longitudinal, and of course you can see apart from the pancreas because it is closely related to the duodenum. The pancreas is present in the concavity made by the C-shaped duodenum, so it is always related to the section of the duodenum. Here we have the junction between the stomach pylorus and the duodenum. Of course, you will notice that it is a junction because there is a change, evident change from one side to the other. Here in the muscle, we have this muscle which is very thick, and here it is relatively thinner. This is the side of the pylorus because this is the thickened inner circular muscle forming the pyloric sphincter. And this is the inner circular outer longitudinal, as you see at the side of the duodenum. If you look at the mucosa, you will not be easily to diagnose the different types of the cells to compare them. No, you will notice that we have white striations all through from the start till the end of the junction. This is different from the gastroesophageal junction, in which we have a part towards the esophagus which is solid, not showing white striations. But here we have these white striations all through because here we have the pyloric glands. The pyloric glands at one side, and here we have villi and crypts at the other side. So you will see in this junction white striations all through. This is the pyloroduodenal, and you cannot see at all the non-keratinized stratified squamous epithelium. So, to differentiate between the two, you will look at the epithelium from the start to the end. If you can diagnose non-keratinized stratified squamous, it is gastroesophageal. If you cannot see it, it is pyloroduodenal. Not only that, but also that the striations, white striations present all through the mucosa denotes the pyloroduodenal, while in the gastroesophageal I have a solid part with no striations in the epithelium and a part which is interrupted by the white striations of the fundus. So the other slide will be the gastroesophageal. Here we have a thick muscle and thinner muscle, the same in the gastroesophageal, so the only clue is by looking at the mucosa. Here we have mucus glands in the duodenum as we have mucus glands in the esophagus. So the easy thing to differentiate between the two is looking at the epithelium. Here we have the first slide in the trial exam. Identify the structures 1, 2, 3, 4. Number 1. Where is number 1? This is 1. Here 1, it is a fold of the mucosa. So it is a plica, not plici. Plica, it is singular. Plica circularis. It increases the rate of absorption here 3 times. Number 2, here, number 2, these are finger-like projection, so these are the villi. They increase the absorption here 10 times. Then we have number 3, here it is all of the mucosa formed of villi and crypts. Number 4, it is here connective tissue of the submucosa. Why? Because it is after the muscularis mucosa. If we have for the connective tissue here, it is the lamina propria, but here we have the submucosa. And also here, you cannot see the lamina propria clearly because the area is crowded by the crypt and the villi. As you see, it is a lamina propria of the villus, and these are lamina propria in between crypts. Here we have the submucosa after the end of the muscularis mucosa. Number 5, here we have the collection of blue dots denoting lymphoid collection. It is the lymph follicle associated with the gut called gut-associated lymphatic tissue, GALT, gut-associated lymphatic tissue for protection against infections. Here number 2, the blue rectangle surrounds the villus finger-like projection above the surface. Number 2, number B, yellow shape surrounds the crypt downward invagination here, which is the intestinal crypt of Liberkin. Number 3, green line indicates here it is a connective tissue of lamina propria here in between the crypts, here inside the villus. Number 3, the black arrow denotes the muscularis mucosa. Slide number 3, we have in the shown intestinal villi, name the cells outlined by the black. Here I have columnar cell with brush border, so these are the enterocytes or absorptive columnar cells. What is the function of these cells? They are responsible for the termination of digestion, yani complete the digestion of carbohydrates and proteins, and also they are responsible, of course, as their name denotes, the absorption of digested material. The absorbed material will pass to the lamina propria of the villus. Here we have the blue circle, which is the goblet cell, which have an upper expanded part and lower cylindrical part containing the triangular nucleus. The upper part is full of dissolved mucus, and it is responsible for secretion of mucus for protection and lubrication. Number 4, the red circle here, it is the villus. Of course, you know it increases the absorption in the intestine by 10 times. while the brush border increases the absorption by 20 times. Here we have identified the layers marked by the red star. We have a black circle also. This is the intestinal crypt. What the type of the gland? It is simple tubular gland. And of course, you know that it is lined by many types of cells. The most important is the Paneth cell at the base of the cell to secrete intestinal enzyme and lysozyme. Here we have the layers marked by the red star. Here, the red star here, it is the lamina propria of connective tissue, and the black star, it is the muscularis mucosa, of course. This is the muscularis mucosa on which the crypts are sitting down. Number 5, name the structures that stain positive with PS. Of course, this is the goblet cell because the PS stain the mucus as it is a glycoprotein. So these are the goblet cells. Star here, and these are the goblet cell, and arrowheads, these are also the goblet cells. But here the arrow, these are the goblet cells in. Here the arrowhead, this arrowhead is the brush border of the enterocytes. So these are the goblet cells. This is the lamina propria, and these are the goblet cells. This is the eosinophilic brush border of the enterocytes, which are columnar cells with basal oval, and this brush border increases the absorption 20 times. These cells, of course, they are columnar absorptive cell. Number 6, this is a section showing intestinal crypt. Very nice. Number 1 here, these are vacuolated cells, they are the goblet cell. Number 2, here it is the lamina propria of the mucosa present in between the crypts. Number 3, it is the lumen of the intestinal crypt of Liberkin. Number 4, it is the Paneth cells at the base of the cells, at the base of the crypt, and it is pyramidal with basal rounded apical acidophilic granules, and you should know the function of these cells. Number 5, it is the muscularis mucosa. Here also this is a section showing the lower part of the intestinal crypt. Very nice. These are the goblet cells, and these are the Paneth cells. This is the brush border of the enterocytes. Here identify the layer marked by the red line. It is the muscularis mucosa. The encircled cells are the goblet cells. The cells marked by the A, it is the Paneth cells, responsible for secretion of intestinal enzymes and lysozyme. Number 8, identify the organ here. It is provided with villi and crypts, so it is a small intestine. And you have these mucus glands in the submucosa, so this is the duodenum. And you should mention that because the question is asking for the organ in a specific way. Number B, name the structures marked by the black stars. These are the villi. Describe their shape. They are here. The shape of the villi in the duodenum is characterized by being broad leaf-like. Broad leaf-like. Very important. Here, name the structures marked by the red star. It is the Brunner's glands. Mention the function. They secrete the alkaline mucus to neutralize the acid chyme to protect the duodenal mucosa, of course, and you know also that they decrease the secretion of the gastric HCl. Here, this is the ileum, will not be given now. Identify the organ, be specific. This organ is provided with villi and crypts, projections, downward invagination, and mucus glands. So it is never esophagus at all. It is the small intestine because we have the villi and crypts, and these are the Brunner's glands. This is the musculosa, and this is the pancreas. Identify the organ, be specific. Duodenum, green arrow, it is the villus. A red arrow, it is the crypts. Yellow arrows, muscularis mucosa. Black arrows, this is the Brunner's glands. And here we have C, a layer marked by the black line. It is the musculosa inner circular outer longitudinal. And D, it is the pancreas, which is closely related to the duodenum. Number 11, and it is the last slide here. Name the type of the, it is a junction, of course. Type, you will look for the epithelium. It shows white lines all through, so it is not gastroesophageal at all. It is the pyloroduodenal junction. This side A shows this thick muscle, so this is the pylorus, because this is the pyloric sphincter. Side B shows the Brunner's glands. It is the side of the duodenum. The star denotes the pyloric, denotes the musculosa layer, inner circular, that form the pyloric sphincter. The blue arrow points to Brunner's glands. And of course, you know the function of these glands, and they are only present in the duodenum. Thank you very much.